Hi guys. Hopefully we are live now. Um, although it doesn't look like it. I've got a bit of a weird setup today. I'm doing the lesson on one laptop. I'm trying to look at the feed on another one, but that one's not loading. But maybe it's just a glitch with it. Oh, I think we are live. Yeah. Sorry, for some reason it's not loading on my other one. That's weird. Um, Let me see if this will work. Um, oh, that's weird. It looks like I'm gonna have to like I'm just gonna have to press refresh every time I want to see what you guys are saying, but we can work with that. Um, hi hi Hiban, how are you? Um, yeah, where's everyone joining us from today? Oh, also gonna have no video today, unfortunately. I haven't got the webcam, and the way that the computer is set up, the actual like built-in webcam, it would be like pointing straight up at my face from like underneath. I'm writing on it like a tablet style, so it'd be kind of a weird camera angle. So I figured I'd probably spare you guys that, and yeah, just just do it without the webcam. So our session today, we are going through proteins. Um, yeah, probably you guys have already covered this topic. If you started AS this year, it's usually one of the topics that you do quite early on. Most syllabuses they include this in the biological molecules topic. So yeah, you've probably done it before. If not, no worries. You'll be able to like keep up with it today. It'll be a little bit tricky if it's the first time you've seen it, but yeah, it will still be accessible. And if you've done it already, then this will be like a good bit of revision for you. Um, Sweet. You have a test on this tomorrow. Okay, perfect. Um, oh, and Caleb, you're just revising this morning. Okay, yeah, sweet. Well, that's good timing. Uh, this refreshing thing is funny. Okay, anyways, yeah, let's jump straight in. So, okay, a little bit. My name's Alex. I'm the head biology teacher here at Snap Revise, and I've been tutoring and teaching biology for about 6 years now. This is a little bit out of date slide. Um, And yeah, if you've done these sessions before, you'll know that we normally start with a little revision pyramid. So just some questions to get us warmed up on the topic. Could be related to today's like protein topic, or it could just be general stuff to help revise. So first question, not too tricky this one. What molecule is required for a hydrolysis reaction? Oh yeah, I need to refresh. So what do you need to do hydrolysis? I think he wants to do top chat. Okay, nice. For some reason, my thing is like cutting in and out with the chat. So, uh, yeah, you guys are all correct, but there was nothing showing up for a while for me. I was like, I think you guys probably know this one. Yeah, yeah, nice. You need a water molecule, or you could put H2O. Yeah. Hydrolysis and condensation, they're kind of like the opposites of each other, and these come up all the time in the uh, biological molecules topic. So hydrolysis is a like where you're breaking things apart, using water. Condensation reaction is where you are putting, sorry, yeah, you're producing water by forming a chemical bond. So they're kind of, yeah, opposites of each other. Okay, next question. What is an ester bond composed of? And this is the elements basically that you need to form an ester bond. That's what we're asking. Two amino acids. Um, you actually wouldn't get an ester bond between two amino acids. That would be a peptide bond. which we're going to look at a little bit later. And this is actually just the elements. So you get an ester bond in a triglyceride, the formation of triglyceride, or in the formation of a phospholipid. You also get them between nucleic acids. So basically, it's a carbon, oxygen, carbon bond, which kind of overlaps with this one. So a glycosidic bond is actually also a carbon, oxygen, carbon bond. The difference is an ester bond also has a double bond oxygen on one of the carbons. So that's how you can differentiate between those two. So this is the ester one, this is a glycosidic. A peptide bond, which we'll look at later, actually goes from a carbon to a nitrogen directly. So that's a little bit different. There's obviously some hydrogens in there as well. But yeah, slightly different bond there. Cool. Right, so next one. Where can you find protein in the body? What are some of the places to find this? Yeah, nice, man. Yeah, a carboxyl group and an amine group come together. That's your classic peptide bond. Yeah, nice. You definitely get proteins in muscles, and that's probably the biggest source of protein in the body. Although there's also a lot of protein in skin. Skin is the biggest organ, and there's a lot of collagen in skin. Collagen is a protein. But yeah, muscles is probably the first one you think of. And the two main proteins in muscles, actin and myosin, you learn about in the A2 syllabus and how they interact together to form muscle contraction. Muscles, skin, hair. Hair is protein. Well, like some of what hair is made of, keratin is protein. Hair and fingernails. Like the hooves of a horse, that's also keratin. Rhino horn, another type of protein. But yeah, we get proteins all throughout our body. Enzymes are made of proteins, which you obviously find everywhere. Hemoglobin, very important protein to transport oxygen in the blood. So yeah, there are a lot of different proteins in the body. Okay, cool. Next one. How are starch and glycogen different? What would you say the main differences are? See, you could talk about structural differences here, or you could also talk about um, functional differences. So structural differences, you can say that starch depends on your syllabus actually. So if you do AQA, you kind of just say that starch is unbranched. Or they do kind of mention branch starch, but they don't mention it by name. So you definitely don't need to know amylose and amylopectin. But yeah, I guess for any spec, we can say that there is like more branching. So starch is less branched. And in fact, some types of starch, specifically amylose, is not branched at all. Functional differences. So glycogen can be metabolized more quickly. Metabolized faster. And it's more compact. And this is due to its use as the energy storage molecule in animals. Animals need to be able to access their energy more quickly than plants. Plants never need to really have bursts of energy. They don't do anything that quickly, whereas an animal might need to like run for a while, or yeah, probably like movement is going to be the biggest like burst of energy. So they're going to need to access their stored energy much faster than a plant. So they need something that can be broken down very quickly. and turned into glucose, which can then be respired to create energy. So glycogen can be broken down quicker, and it's more compact. Again, this is more useful for an animal than a plant, because animals obviously, well, most animals are motile, so they move around. And if you need to be moving around, the more compact your energy store is, the more efficient you can be. Plants don't tend to go anywhere, so that's less of a concern for them. Cool. Oh, your chat started to work again. What does branched mean? Yeah, good question. So, uh, when you form a polysaccharide, you're basically joining together lots of glucose molecules. So if you join them together in a chain, kind of um like a like a string of beads or something, that would be unbranched. If you add a side chain in there, this would count as a branching. And you can have like branches on branches. So when we say something is highly branched, it's going to have lots of side chains, and those side chains are probably going to have like additional side chains on them. The reason that's useful is because when you want to break it down, you can chop off glucoses from all of these endpoints. Whereas if you had unbranched, there's only two ends you can start to break it down from. So the more branching there is, the more quickly you can break it down and access those glucose molecules. Nice, perfect. So do you guys have any facts about proteins you can share? Or to be honest, we're gonna be jumping to that today anyway. This chat thing. The yeah. I don't know why it's not working at all, but yeah, I'll try and do the best I can. Sign in. Yeah, I might kind of have to do this lecture style, because your comments only load for like a small amount of time, then it stops working again. But we'll do our best. Anyway, so our main objectives today, we're gonna look at the structure of an amino acid. We're gonna look at how you form a polypeptide by condensation reactions, and we're gonna look at forming secondary and tertiary structures. And we're going to go through examples of globular and fibrous proteins. These are the spec points. You should all have a copy of your spec anyway, but if not, you can have a look through specifically what you need to know for your syllabus. For biomolecules, to be honest, they're all fairly similar, but there are some little differences between the specs. Okay, so yeah, this is an amino acid. You guys should all be able to draw out an amino acid from memory. It's not too difficult, and it's one of those things. If you can't do it already, sit down for literally, you could learn it in a few minutes. Just test it a few days in a row, and you'll have committed it to memory for, well, hopefully the rest of your A-level syllabus career at least, and maybe like for longer. Yeah, relatively simple. When you're going to draw it, I would say the easiest way to do it, start with your central carbon. So you just draw a C here. You should hopefully remember carbon forms four bonds. So we're going to draw four things coming off our central carbon. We've got an R group. We'll talk about that a little bit later. We've got a hydrogen. It's nice and easy. Then we've got these two other groups, carboxyl group, C, double bond O, OH. You may recognize this from a fatty acid. Fatty acids also have carboxyl groups. Then you've got your amine group, which is NH2, over on the other side. There's no reason that the amine group is on the left and the carboxyl group is on the right, but for some reason, this is basically, it's always drawn that way around in textbooks. Don't really know why, I don't know who decided that, but obviously it's a molecule, so it could be facing in any orientation, but they just tend to draw it this way around. But I guess if they're trying to be mean, trying to catch you guys out, they maybe would draw it a different way around just to try and confuse you. So yeah, just be aware of that. So yeah, I'm not sure we need to add that much stuff here. I guess amine, you might recognize this, the amine bit from the word ammonification. So it's all to do with ammonia, NH3 ions. Like the same prefix is the same root of the word. So yeah, if you chop off the amine group, you form urea, and that's part of the nitrogen cycle. Which is the NH2 carboxyl group, COOH. And the L group, this is the variable side chain. There are 20 different types, and it's the R group that determines the 20 different types of amino acid. So for all amino acids, these three bits are the same. And it's just the R group that is variable. Perfect. So let's look at how we join these together. So polypeptides are synthesized from amino acids in condensation reactions. So a normal protein will have many hundreds or even many thousands of amino acids. So you're going to be doing a lot of these condensation reactions to form them. So if this is an amino acids, if you join two together, this is going to give you a dipeptide. And they're going to be joined together with a peptide bond. This is actually, this is done usually in the process of translation, which you look at in more detail later on in the syllabus. Oh, I've got your protein fact now made up of amino acids. It's finally loaded. But yeah, it's a bit of a lag. So two, we call a dipeptide. More than two, so these are both obviously more than two. We just call them now polypeptides. So I guess technically you could call them tripeptides, like what would four be? Quad peptide, quaternary peptide. But we basically don't bother. We do dipeptide and then we jump straight to polypeptide. The same as with polysaccharides. You have monosaccharides, disaccharides, and three or more. We just call polysaccharide. Cool, nice. So condensation reactions, we're producing water. So each time we do this, we're going to be producing a water molecule. Sweet, nice. So if we want to break down a protein, which is what, so if you need protein in your diet, we can't produce amino acids ourselves, so we need to consume them in our diet. So you can get proteins from both plant and animal sources. Then when you digest them, you're basically hydrolyzing the protein back down into amino acids. We then use those amino acids to build up our own proteins. So you can kind of think of amino acids a little bit like Lego blocks. You take a bigger piece of Lego that's you're eating your protein. You break it down into the individual blocks, and then you can use those to create your own Lego models, your own protein structures. So hydrolysis reactions, we are putting in water. So you need to break this down into 4 amino acids, we would need to have 3 water molecules. essentially one to break every peptide bond. And for 4 amino acids, we're going to have 3 peptide bonds to break. Nice. Yeah. So I guess, well, if you want, let's draw out what that would look like. So proteins bonded together would look, you've got a carboxyl group. In fact, let's do it. I'll draw one without first. So this is not bonded. So I haven't drawn the whole amino acid. Obviously, there's like, this is your central carbon, so there's more stuff coming off there, but that will just give you the idea. So this is where you're going to form the peptide bond. We remove, oops, I've missed a water in there, oxygen. So we remove water. So it's the hydrogen from the amine group and the OH from the carboxyl group that gets removed. And then you'll see we are left with this carbon, double bond oxygen, nitrogen, hydrogen, and then the central carbon of the other molecule. And we get plus H2O. So this way is your condensation. Going this way is your hydrolysis reaction. Relatively easy to do. And yeah, happens all the time when we break down proteins and recycle the amino acids. We also recycle amino acids in our own bodies. So like the lysosomes in the cell, they will break down old or obsolete proteins or enzymes, and the amino acids can be reused for other purposes. So yeah, we need them in our diet initially, but we can also reuse our own amino acids as well. And you can respire amino acids. The body's preferred source of preferred respiratory substrate is glucose, but it's perfectly happy respiring proteins as well. The only downside is this amino group. So you're going to be producing more urea, but it is also an option for respiration. Okay, cool. So hopefully you guys are following along so far. And yeah, I think we're going to do some questions. Yeah, perfect. So have a look at this diagram. And the first question, so it's just a 1 marker, so it won't be too complicated. State the chemical symbol of the atom represented by letter X in figure 1. And again, I'm probably not going to be able to see what you guys are saying until maybe a little bit too late to be useful. Oh, I got your one there. Yeah, nitrogen, exactly. Nice. So chemical symbol. So actually don't write nitrogen, you just write N, correct. Name the polymer formed of a chain of amino acids. So there's 2 things that would be accepted here. One I would say is slightly better than the other one. Let's see what you guys come up with. Oh, my chat's disconnected anyway. So can't really see what you're saying, but hopefully you said one of the 2 things. The better answer would be polypeptide chain. Well, the chain bit is actually kind of optional. So polypeptide is enough by itself, or if you wanted to add chain, that's okay. They maybe would let you get away with protein, but that's a bit dubious. I would say if you said primary structure of a protein, then they'll be more likely to accept that, because a polypeptide chain is the primary structure of a protein. It's not actually the same as a protein. So yeah, I would definitely recommend you go with this one. Cool. And then name the bond formed when 2 amino acids joined together. Describe the formation of this bond. So name of the bond is a peptide bond. And description of formation. You could say something like amine group, or amino group actually. Amino group joins with carboxyl group. Or you could say, I don't have that much space, but even better would be to say the amino group of one amino acid joins with the carboxyl group of a second amino acid, just making it a little bit more clear. And I would mention water. So something like water is formed. Cool, nice. So let's look at the structure of a polypeptide in a little bit more detail. So we need to basically, you go through the different levels of protein structure from primary all the way up to quaternary. Primary structure, relatively straightforward. This is basically the sentence you need to memorize. So the primary structure is the either sequence or order of amino acids in the, I would maybe actually change that to polypeptide. Order or sequence, that doesn't really matter, but polypeptide is better than protein. And the bonding in that. So all these bonds are peptide bonds. And this is basically formed during translation. Nice. Then we're moving on to the second level of folding. So we call this the secondary structure. There's two main shapes here. We've got beta pleated sheets and we've got alpha helices. This is the alpha helix. Sometimes you see it drawn like this. The Greek letter alpha and then helix. And sometimes you see it drawn like this. The Greek letter beta and then just pleated sheets. They are both held together by hydrogen bonds. Hold structure together. And yeah, the alpha helix kind of gives a bit of a clue. It's a spiral structure. The beta pleated sheet is a little bit like, you know, if you fold a piece of paper into a fan, or if you just get one of those fans that like opens up, it's kind of the same, well, similar structure to that. Yeah, this sort of like folded pleated shape. So you get those shapes starting to form, and they come together and they fold up in sort of three-dimensional space to give rise to the tertiary structure. So this is the one that's most associated with the shape of the proteins. So you can see here, like these sections, this and this, these are alpha helixes. And this bit here, this is a beta pleated sheet. So you can see how they're sort of like folding up around each other in three-dimensional space. So this is like tertiary determines 3D shape. And there are 4 types of bonding that are involved with this. You've got hydrogen bonding. Same as the secondary. You've got ionic bonds. You've got disulfide bridges. And you've got these things called, they're not technically a type of bonding, but they do help hold the structure together. You've got hydrophobic and hydrophilic interactions. So hopefully you recognize these words maybe from the looking at phospholipids, which have hydrophobic and hydrophilic parts. Hydro means water. Phobic and philic means like loving and hating. So hydrophobic doesn't like water. Hydrophilic does like water. Maybe the word like isn't the best. Like probably something like interacts well with water or doesn't interact well with water would be better. So this is a property that's particularly important in globular proteins. Globular proteins, we're going to look at them later. They're the sort of 3D spherical round proteins. Things like hemoglobin or amylase, basically any enzyme, are going to be globular. Their 3D sort of spherical shape is maintained partly by hydrophilic amino acids facing out, hydrophobic amino acids hiding away in the middle, just like in the phospholipid bilayer, where you've got your hydrophilic heads facing out to the water and your hydrophobic tails all hiding in the middle of the bilayer away from the water. It's kind of the same thing with proteins. So the hydrophobic R groups in the middle, hydrophilic R groups on the outside. Nice. So some proteins, to be fair, not many, but some, they finish at the tertiary structure. So that is the biggest that they get. But most proteins will actually have a quaternary structure. So this is where you have more than one polypeptide chain coming together. So for example, hemoglobin has 4, collagen has 3, a blanked on their name, an antibody, that's what I was looking for. They've got 4. So yeah, most proteins have more than one. Some have many. Like for example, ribose in plants has something like 23 or 24. So you can have quite a lot. The bonding, this is the same as tertiary structure. So the same 4 types of bonding there. And yeah, this basically allows you to create bigger, more complex proteins by joining together different polypeptide chains. You can also add other bits. So for example, you could add like a carbohydrate chain to turn it into a glycoprotein, or add a lipid to turn it into a lipoprotein, or yeah, add an OH group to make it more water soluble. You can further modify that. That's one of the things that the Golgi does. If you remember, the Golgi is involved with modification of proteins. Cool. So we're going to look at some of those bondings in a little bit more detail. So hydrogen bonds, these are the weakest of the bonds that hold everything together. They are just attractions between the oxygen and the hydrogen. Basically, the oxygen is slightly negative. Hydrogen is slightly positive. And hopefully, as you know, positives and negatives attract. So just like on a magnet, you have the positive end and the negative end. It's not actually magnetism, but it's the same idea. So positives slightly attract negatives, but because they're only slightly positive, slightly negative, this attraction is relatively weak. So you may, or hopefully you've seen these letters, delta. So we say oxygen is delta negative, and hydrogen is delta positive. And that just means kind of like a weak attraction. We won't go into the actual full details of what causes that, but it's to do with how good they are at attracting the electrons in the covalent bond. That's going into a bit too much detail for today. If you do chemistry, you'll probably be super familiar with that anyway. Oxygen and the hydrogen here. Cool. So an ionic bond. This is a bond formed, well, probably not too surprisingly, between two ions. So they have to be oppositely charged ions. Oh, sorry, I've just seen your question. I don't know how long ago you asked it. What is the difference between peptide and ester bonds? Yeah, so it's just the molecules that are in the bonds. So you get ester bonds between a carboxyl group and an OH group. So this is a carboxyl group, and let's just say, what can we have here? Let's just have a carbon with a simple OH group like that. So an ester bond would form between these. So an OH group interacting with another OH group, but crucially, because that could be a glycosidic bond, crucially you have to have this for it to be an ester bond. Whereas the peptide bond is between two amino acids. So this is the one we drew on the other page. This is your peptide bond. So you have a carbon bonded directly to a nitrogen. That carbon also has a double bond oxygen. The nitrogen has a hydrogen. So yeah, hopefully that answers your question. And apologies, that was like 10 minutes after you asked it. Like I said, the the chat for me is loading like very slowly and not at all in real time. So yeah, ionic bonds form between oppositely charged ions. So what should we say? Like positive ions attract negative ions. And these are the next weakest type of bonds. So they're stronger than hydrogen bonds, but they are weaker than full covalent bonds, which disulfide bonds are an example of. So these are covalent. They're strong. The strongest type of bonding in the tertiary structure. And they actually only form between one type of amino acid. So cysteine, this is an amino acid. Yeah, if two cysteines come near each other, they have this element sulfur, and the sulfur can form a bond between the two sulfur molecules. This is your disulfide bond. You get lots of this in keratin. So that's the molecule that forms a bit of your skin, your hair, and your fingernails. They're all made of a lot of keratin, and sulfur is a very strong smelling chemical. So when you burn hair, you're releasing all that sulfur, and that's why it has a really strong smell. Like the smell of burning hair is like quite acrid. That's that sulfur being released. Anyway, it's not super important for biology, but the biology A level, but yeah, just have interest. And this is the final bit: hydrophobic and hydrophilic interactions, the final type of bonding. This is a diagram kind of showing what I was talking about before. So this is your polypeptide chain. You can see they've colored the hydrophobic and the hydrophilic regions in different colors to show how this is going to interact with water. So we've got water all around. H2O, H2O. So you can see the hydrophobic areas are hiding away from the water. The hydrophilic areas are sort of all facing on the outside. So this is mostly in globular proteins. Globular proteins, yeah, it's not technically a type of bonding, but it does give rise to the structure, but it's not a specific bond per se. Cool. So yeah, hopefully you guys are still following on, and yeah, let's look at some questions. So describe how the R groups can determine the tertiary structure of a protein. Cool. So it's 4 marks. Basically, they're going to want you to talk about the bonding in tertiary structures. So we could basically go through the different R groups. So ionic bonds can form between oppositely charged ions. Hydrophobic R groups on the outside of molecule. Hydrophilic on the inside. Sorry, other way around. Hydrophobic on the inside. Hydrophilic on the outside. And then for the final mark, just mention the disulfide bonds. So disulfide bonds can form between cysteine R groups. And it's only cysteine for that one. Nice. So we're now going to look at some specific examples. So the case studies that they use in the specs most often are collagen and hemoglobin. This is one example of a fibrous protein and one example of a globular protein. So let's look at collagen first. So this is our fibrous protein example. Its function is to give structure and strength to, you find it in the skin, in tendons, it's also in bone. So it's the main building material of the body, or the main building protein of the body. Its structure is, it has 3 polypeptide chains. And they are sort of intertwined to give strength. So kind of like how you can knit together many cellulose fibers to give rise to a stronger, like bigger strand. Same as this, like these 3 polypeptide chains, they're made stronger by wrapping them all together. And there's some attraction between the different strands, keeping them in this formation. So it's kind of like a plait, which is strong. Fibrous proteins tend to be insoluble, and collagen is an example of that. This is because they need to be, they're building materials, so you don't want them dissolving in an aqueous environment, because you need the like if you built a house out of soluble materials, your house is just going to dissolve in the rain, and the body is obviously a very aqueous environment. So if you're building things out of materials that are soluble, they're just going to dissolve, and it's not going to do its job. So they tend to be insoluble. They're usually large molecules. Again, collagen is a large molecule. And they tend to have lots of amino acids kind of in a repeating structure. Collagen in particular has a lot of glycine, proline. These are both very simple amino acids. Their R groups are quite small. And that basically means that there's not a lot of R groups getting in the way. So they're basically just a long chain of like carbon, carboxylic acid to amino to carbon, carboxylic acid. There's not like, they don't have big R groups. And so it helps them to sort of plait together a bit closer and work better as a structural molecule. hemoglobin. So this is the molecule that transports oxygen in the blood. This is a very important molecule. In fact, it has its own whole little chapter that you learn about it in more detail. It's made up of 4 polypeptide chains. Some specs split this up, so you've actually got two alpha chains and 2 beta. So it's basically just like, yeah, 2 different types of polypeptide chain that are coming together. And each of those polypeptide chains has a heme group. And this is what transports the oxygen. And this is your heme group here. What actually allows us to transport oxygen is the presence of an ion. So it's an ion, kind of a weird thing to say, Fe2+. So Fe is a symbol for ion, like iron, like that. So it's an ion. Yeah, this. Cool. Then yeah, hopefully that's you guys are following along so far so good. And let's try some questions. I think there's the last bit. Cool. Yeah. So we'll do these questions, and then we are done. So we've finished. Well, it's gonna be a little bit less than an hour. I guess we just got through it very efficiently. Do we need to learn details of disulfide bonds? Yes. Not like you don't need to be able to draw the structure of what they look like when they're bonded together. You just need to know that when you have one sulfur and another sulfur here, so they'll be bonded to a carbon. They just basically, in fact, you don't even need to know this much detail. You just need to know that the sulfurs can bond with other sulfurs. This is a covalent bond, and therefore it's strong. So if you know that it's a covalent bond formed between sulfurs, and those sulfurs are only found in cysteine, that is everything you need to know about disulfide bonding for biology. Yeah, literally there's like covalent, strong, only in cysteine. That is enough information. But yeah, good question. Okay, state, whoops. What's happened here? Okay, no, back. State one function of hemoglobin. So yeah, there's obviously one main one, but it does. Maybe it has some secondary functions. Actually, it does have secondary functions. So maybe you know another one, but it has a big one. So yeah, nice. The main one is obviously it transports oxygen. Transports oxygen, brackets, in the blood, if you want to add that. It has another role in the blood. It actually helps to maintain pH of the blood. Or I suppose you could also say it transports carbon dioxide. Transports CO2. It's actually not the main way that CO2 is transported. The carbon dioxide, the waste product that we get from respiration. This is what we're using. We're using the blood to take this to the lungs to remove it. Whereas we use the hemoglobin to take the oxygen from the lungs to the respiring tissues. Most CO2 is not transported by hemoglobin, but a small amount, but around roughly like 5% is transported by the actual hemoglobin. So that would be a possible answer as well. But yeah, I would basically say you're taking an unnecessary risk if you just didn't say this one. So just go for that. Okay, hemoglobin contains a prosthetic group known as heme. Collagen does not contain a prosthetic group. Describe 3 other ways in which the structure of hemoglobin differs from that of collagen. So this is a classic type of question. Basically, it's asking you to describe differences between globular and fibrous proteins, but they've given you specific examples. So what can we say on this? Prosthetic group, good question, yeah. Prosthetic group is a non-protein section of a protein, and it is permanently attached. So that the permanently attached is just to differentiate it from a coenzyme or a cofactor. So the heme group has got an ion in it, an iron, which is not part of the protein. So it's a non-protein section that's permanently attached. Apologies for the coughing as well. I've had my first cold of the winter. Just kind of I've almost shaken it off now, but the last like week been a bit yeah, been suffering from this cold. Hopefully the first and maybe hopefully the last, but we'll see. So yeah, things we can say. Differences. So collagen has 3 polypeptide chains. I'm just going to put PPC, but that's not the actual proper acronym. Whereas hemoglobin has 4. Hemoglobin is soluble. Collagen is not. Collagen is, what should we say, long or rope-like or fibrous, whereas hemoglobin is spherical. Should I refresh this in case you guys put anything different as well? Check. Yeah, no. So any of those 3 would have been suitable. What else could we have maybe put? Yeah, no, those are probably the best ones. You could maybe talk about the type of bonding. Like hemoglobin's going to have hydrophobic, hydrophilic interactions, whereas collagen doesn't really have that. But I'd say that's a weaker point than the other ones that we have. So I'd probably just go with one of those. Yeah, nice. And that's it. So hopefully you now know the structure, the levels of bonding in a protein, and know a little bit more about hemoglobin and collagen specifically as examples. If you guys have any questions, please feel free to ask. Otherwise, yeah, thanks for joining the session. Hope it was helpful, and see you guys in the next one. We do these every month. So yeah, and we have other subjects. So if you do maths, chemistry, or physics, we also have lessons in those. And if you enjoyed the session, we have a website where we host these sessions on a weekly basis. So there's a lot more content. So it's actually a subscription model. But if you wanted to do some more sessions, then that's a really good way to supplement your revision and your learning process in school. Yeah. So yeah, have a good rest of your Sunday, and yeah, see you hopefully next month. Cool. Bye guys.